So welcome everyone, and thank you all for attending this monthly Dunlevie Center lecture series. I'd like to just start with some updates, and then Virginia will introduce our guest for today. Our fall 2023 seed grant awardees were selected, and congratulations to them, to Andrea Henkel for her randomized controlled trial proposal of Cabergoline, if I pronounce that correctly, for lactation inhibition after early second trimester abortion or pregnancy loss, the ALISTA study. And Dr. Christy Redhorse creating a 3D atlas of placental perfusion over gestation. These were wonderful proposals, very competitively vetted. So congratulations to this cycle's awardees. Our next center lecture is on January the 11th, and it'll be our guest lecturer is Dr. Sarah Osmundson, who's Vice Chair of Research and Associate Professor of Obstetrics and Gynecology at Vanderbilt, and she'll be talking about predicting post-discharge opioid use after childbirth, matching prescribing to actual use. Sarah completed her maternal fetal medicine fellowship here at Stanford, and so it'll be very exciting to have her return as a guest lecturer. And Virginia, I will hand things off to you.
Wonderful. So it is my immense pleasure to introduce our speaker for today, Dr. Sam Mesiano. He is the William H. Weir Professor of Reproductive Biology in the Department of Reproductive Biology at Case Western. And he also serves as the Vice Chair of Research for the Department of Obstetrics and Gynecology, and also has an appointment as Professor in the Department of Physiology and Biophysics. He received his PhD from Monash University and has been involved in understanding progesterone and steroid hormone biology as it relates to human parturition and pregnancy. His first paper that he published in 2002 was progesterone withdrawal and estrogen activation in human parturition are coordinated by the progesterone receptor A expression in the myometrium. And his work really has been instrumental and essential in understanding parturition and how human parturition is different in its mechanism of how it recognizes progesterone withdrawal, since in humans, progesterone levels don't go down as it does in animals. That was so nicely demonstrated previously. He's then completed postdoctoral fellowships at UCSF, where he was involved with adrenal gland studies. So I assume that was with Dr. Jaffe. I have known Sam, I think, primarily through the Society of Reproductive Investigation, or when we met was called the Society of Gynecologic Investigation. And we've both served on the Leadership Council, and he also serves on the board of the Preterm Birth International Collaborative. I always enjoy seeing his elegant and rigorous science. Obviously, parturition and preterm birth is an important topic for this group. And so I look forward to his sharing with us his expertise and the advances he's been able to contribute to. So thank you, Dr. Mesiano.
Right. Thanks, Virginia. Elegant? I'm not sure about that, but thank you for that kind introduction. I am going to share my screen and we will barrel into this. Okay. So I'm assuming you can see my first slide.
You're currently in presenter mode, so I think if you can do the
me.
swap. Yep, that's perfect now.
Okay. How about this? Good.
Beautiful.
Good? Okay. Let's start with no disclosures. There you go. What I'm going to do today is mix a bit of basic research with some clinical relevance. I know most of you out there are clinicians, and I'm going to talk about the story of progesterone in a sense. And as you all know, progesterone is called progesterone because it's progestational, and it was posited as being a, therapeutic strategy to prevent preterm birth. Recently, in the last 20 years, 17-hydroxyprogesterone caproate delivered intramuscularly to women at risk for preterm birth has been a standard of, not standard of care, but it was used widely. Vaginal progesterone is currently used. And so the question of whether or not we can exploit progesterone to prevent preterm birth, I'll talk about that. And, in order to actually do that, we need to understand the mechanism of progesterone action on the gravid uterus and how its actions are regulated to allow for parturition. And then I'm going to end with talking about some work we've been doing in the last 10 or 15 years on exploiting the actions of progesterone to develop new therapies, new therapeutic strategies to prevent preterm birth. I'm sure I don't need to go through this. This is the current clinical guideline from ACOG regarding vaginal progesterone. It can be used as a treatment option for patients with a history of preterm birth with a short cervix. But the intramuscular 17-hydroxyprogesterone is no longer recommended. And there is some argument as to whether vaginal progesterone even works. So the point I'm going to make through this presentation is that why did we think progesterone would work in the first place when women are marinating in progesterone for most of pregnancy? And there's no evidence that there's a progesterone deficit that is increasing risk for preterm birth. Nonetheless, clinical trials were done and clinical practice was changed. More clinical trials were done, and we're back at the same place where we're still uncertain whether or not progesterone can be used to prevent preterm birth. And so I want to move forward from that and drill down into the mechanism of how progesterone acts in order to possibly rethink the use of progesterone as a therapeutic to prevent risk or decrease risk for preterm birth. So this we all know, that progesterone exerts a block to labor, then something happens that removes the block, and that allows the uterus to transition to the laboring phenotype and the full then cascade of events leading to birth. In most species, you can see here the trajectory of progesterone levels during pregnancy and estradiol levels in the green, progesterone in the red, showing that prior to the onset of labor, in most species, there's a decrease. And I'm assuming you can see my cursor, is that right?
Yes, we can see the cursor.
There's a systemic fall in progesterone that precedes the onset of parturition. And in these species, we call them species that are dependent on a systemic progesterone withdrawal. But there are others, like the guinea pig, where there's no systemic progesterone withdrawal. And as Virginia alluded to, in women, there is no systemic progesterone withdrawal. Yet, if you interrupt progesterone signaling in women, then it induces changes that are that will lead to the onset of labor. So there's a conundrum here. If progesterone is important for staying pregnancy and removing progesterone's actions are important for going into labor, how are progesterone's actions removed in order to allow labor to happen in women? And this is what we've been studying for the last 20 years. And so the mechanism of progesterone action is typical of all steroid hormones. It binds to a receptor protein, then dimerizes. The dimer with the progesterone bound translocates to the nucleus, where the progesterone receptor acts as a ligand-activated transcription factor to regulate the expression of a bunch of genes. And if this is in a myometrial cell, then the phenotype that happens in response to this signaling pathway is uterine quiescence, myometrial relaxation, and the continuation of pregnancy. But when this is interrupted, for instance, with RU486 or mifepristone, a progesterone receptor antagonist, then in pretty much all species examined, viviparous species examined to date, this will cause emptying of the uterus, the ending of pregnancy by the process of labor and delivery. The other thing we know is that pro-inflammatory stimuli acting predominantly through the NF-kappa B pathway can also drive the expression of genes that are pro-labor and can induce labor. And we know this from animal models that I'm going to talk about later on in the presentation. And it's clear that the process of labor and delivery involves an inflammatory event at the maternal-fetal interface and the tissues of the gravid uterus. So the way we think about this is that for most of pregnancy, progesterone somehow exerts a blocking effect on these drivers to move the uterus into the laboring state. And we refer to this as the progesterone block. And that the trigger mechanisms for parturition then interrupt this block to allow this default pathway or this knee-jerk type reaction of the uterus emptying to take place. Now, I think of the uterus as being an emptying organ. It's during labor on a per weight basis, a smooth muscle of the uterus, the myometrium, is one of the strongest muscles in the body. So for 40 weeks, it's being fooled into thinking it's a storage organ. Works great as a storage organ, but it works really well as an emptying organ. So how does this work? How does progesterone do this? Well, I'll just go back one slide. We were focused on these mechanisms by which the parturition trigger signal can interrupt this signaling pathway of progesterone through the progesterone receptors. And so the first thing we looked at was the progesterone receptors themselves. So this is a somewhat busy slide, but what it shows is that the progesterone receptor is encoded by a single gene that makes two proteins from two different promoters, the full-length PRB and the truncated PRA. And most of the transcriptional activity of progesterone is mediated through PRB. Now, both proteins have the typical elements of a steroid hormone receptor with a ligand binding domain, nuclear localization signal. Both can be phosphorylated at many places, and both affect transcription of different genes. And there's an overlap in the set of genes that these receptors will regulate in response to ligand binding, in response to progesterone. But one of the interesting things is that PRA acts as a repressor of the transcriptional activity of PRB in most cell types. So this sort of led us to think, well, what if that withdrawal of progesterone signaling in parturition may be via an activation of this transrepressive activity of PRA? And so these are old data, and one of the first things we did then was look at the levels of PRA and PRB, and these are Western blots where B is bigger than A, so it runs a little higher. In myometrium from women who are having a C-section, either preterm and not in labor, or at term and either not in labor or in labor. And if you distill all the data down, what we see is that the onset of labor is associated with an increase in the level of PRA in the myometrium, which is consistent with this dominant negative effect of PRA beginning to take hold and preventing the pro-pregnancy actions of progesterone to continue, effectively causing a functional progesterone withdrawal. And much to our satisfaction, these data were replicated by others many years later. So we then looked at this effect with cells on a dish, and sure enough, when you take myometrial cells and you put them on a dish and you expose them to an inflammatory stimulus, they do make more of the predicted proteins. One of them is IL-8. You can see on this Western blot here that IL-8 goes up in response to IL-1 beta. But now we can we can modulate the levels of PRA and PRB. So if we have the cells tuned in for a PRB dominant state, then this ability for IL-1 beta to increase IL-8 is significantly repressed by progesterone, okay? So this is an anti-inflammatory effect of progesterone, or a pro-pregnancy effect of progesterone, effectively blocking these pro-inflammatory drivers. But if we then switch the cells to a PRA dominant state, then this anti-inflammatory effect of progesterone is lost. So this was some nice in vitro evidence, at least, that there's a functional consequence of increasing PRA levels, in that you lose this pro-pregnancy effect of progesterone. We also, and I won't show you the data, more recently found that this phosphorylation event, one in particular at the 345 position of the two proteins, when this is phosphorylated, the ability of PRA to trans-repress PRB is lost, and so, or is gained, actually, I'm sorry. And so this is also a critical step in possible mechanisms for withdrawing the actions of progesterone to allow parturition to happen. So our working model then is that we have two counteracting pathways. We have progesterone acting through the PRs to exert a block, and then we've got the pro-labor, pro-inflammatory stimuli that are hitting the uterus and trying to push the uterus into the laboring state. Now, for most of pregnancy, the progesterone PR pathway dominates, and we end up with a relaxed uterus. But then what we think is happening is that these stimulatory pathways reach a threshold whereby they start to modulate the capacity for the progesterone receptors to transduce this signal and cause a loss of the pro-pregnancy actions. And so the way we're thinking about this is that pregnancy is a stress on the female physiology, and that stress impacts an inflammatory load on the uterine tissues, so that as pregnancy advances, you can imagine this inflammatory load to increase until it reaches a threshold. And once it reaches that threshold, then the functional various mechanisms of progesterone progesterone withdrawal are activated, and that then triggers the cascade leading to parturition. Okay, so our question then is, well, can we manipulate this threshold? And you can imagine that in a pathologic situation where you've got all these inputs being somehow altered by, for instance, intrauterine infection, or there could be social determinants where there's a lot of stress in the environment. Uterine distension could play a role if you're carrying twins. Fetal development might be affected, and that might send signals that are all integrated into this inflammatory load. So the shifting of this curve and the shape of this curve, and when it reaches this threshold, will determine when parturition happens according to the x-axis, which is time. And so preterm labor is a multifactorial etiology that it's understandable if you think about all these different factors that could contribute to the inflammatory load. And the way we think about this is that once this is activated, then the progesterone withdrawal mechanisms kick in, and then the chocks are taken off the wheels, and the 747 can go barreling down the runway, leading to labor. Okay, so is there any clinical data to support this? Well, I just want to show one experiment that will never be done again. This was in 1958 when Upjohn, the drug company, 10 years before, had produced a lot of progesterone and synthetic progesterone, pure progesterone. They were giving it out to people all over the world and saying, do stuff with it. Let's see if it has any clinical utility. The first thing people did was give it to women to see if it would prevent preterm birth. It didn't. And I can go on the whole rant about those data and why we actually did the 17-hydroxyprogesterone caproate study in the first place. Nonetheless, at a meeting in Upjohn in Augusta, Michigan in 1961, Upjohn brought all the people that were working on progesterone at the time together and said, tell us what you got. And this paper by Lars Bengtsson was highly, it impacted me, because with this paper, what they did was they took women who were electing to have a termination at between 12 and 20 weeks of gestation. And the way they were doing the terminations at that time in Sweden was to either do membrane rupture, an amniotomy, and introduce an infection into the uterine compartment. The women all developed a fever, and within a week, they all went into labor and delivered the conceptus. The other way they did it was to inject into the amniotic fluid formalin and effectively cause demise of the conceptus. And in this situation, there was no inflammation, but the outcome was exactly the same. They both emptied their uterus, both groups, within 3 to 7 days. In the group that got the membrane rupture and the intrauterine infection, the circulating the levels of P4 were unchanged as measured by urinary pregnanediol. Whereas in the group that got the intraamniotic infection, the progesterone levels declined because the placenta was killed. So the placenta could no longer make any progesterone. Both cases caused labor. So Bengtsson took this progesterone that was provided by Upjohn, gave 200 milligrams IM every 48 hours, beginning 2 days before and continuing for 4 days after the women in this experiment. Okay, so some women got progesterone, others didn't. The ones that got progesterone, okay, if we look on the intraamniotic side, the ones that got progesterone never went into labor. Okay, so by replacing the progesterone that was lost because of the intraamniotic formalin, pregnancy, the uterus maintained the relaxed phenotype and stayed pregnant. And in these women, they had to stop giving the progesterone, and within days of not giving the progesterone, they all emptied their uterus. In the women that got the membrane rupture and were infected, they still went into labor despite the high levels of progesterone. So what this is telling us is that inflammation overrides the progesterone block, and that inflammation somehow causes progesterone withdrawal. So that fits with our model that these inflammatory pathways that are trying to tell the uterus to empty are in fact also causing the progesterone withdrawal when they reach that threshold that I showed in the previous slide. So our next question was, well, how does this happen? And this just summarized what I just said. As those inflammatory pathways are reaching the threshold, they change either the receptor or, I'll go into some potential pathways for this. The bottom line is that inflammation induces progesterone withdrawal. Okay. Now, all right, so if this is the case, where can we intervene? Well, we could think of, maybe increasing the threshold so that this ability for the inflammatory load to induce progesterone withdrawal is somewhat desensitized. We can block the signaling pathways that cause the inflammatory threshold to cause progesterone withdrawal, or we can boost the actions of the endogenous progesterone, or we can do all three. But the key is to move this trajectory to the right and have the natural progesterone continue to work until the pregnancy reaches term. So what I'm going to tell you next is some experiments we've been doing to look into this possibility of interrupting this pathway by either boosting the actions of progesterone or inhibiting this inflammation-induced progesterone withdrawal. So we can think about this as there being two pathways through which the inflammatory stimulus could interrupt the actions of progesterone. The first one we looked at, and I'm not going to show you any data on this, I alluded to it earlier, but basically what we found was that if we take myometrial cells and we expose them to an inflammatory stimulus like IGF, interleukin-1 beta or LPS, then these cells will phosphorylate PRA, and that will activate the capacity for PRA to block the actions of PRB. So that phosphorylation is what we think a key mechanism for this functional progesterone withdrawal. And so we're looking at different ways that we can potentially interrupt that. On the other side of the coin, and more recently, data has emerged to suggest that the cells, the steroid responsive cells, the progesterone responsive cells in the uterus express a gene by the name of aldoketoreductase 1C1, AKR1C1, that has enzymatic activity which will take progesterone and inactivate it and convert it to 20-alpha dihydroprogesterone that is inactive. And this is analogous to testosterone being converted to dihydrotestosterone by androgen responsive target cells. But this is the opposite. It's inactivating the progesterone before it gets a chance to occupy the receptor. So this AKR1C1 mechanism is like a gatekeeper to prevent the endogenous progesterone from actually activating the receptor. So you can imagine that if we can develop selective progesterone receptor modulators that might bind the progesterone but not allow for the phosphorylation, then we can maintain the block to parturition. Or if we inhibit the activity of this enzyme by a selective progesterone receptor modulator that is not a substrate for this enzyme, but is a potent progestogen, binds the progesterone receptor and maintains pregnancy, then we could potentially shift that curve to the right. So we did a series of experiments, and the experiments I'm going to tell you next focus on the maternal fetal interface, which really is the fetal membrane periotalis, where you've got the amnion, the chorion, alongside it, the decidua, and then alongside that, the myometrium. And we're interested in this interface because this is where the maternal immune cells sit that become activated as part of that inflammatory driver for inducing parturition. And it's also right next to the engine for birth, the myometrium. So we think that this is the area of the maternal fetal interface that is the key leading edge to determine whether or not, and I'm using an aeronautic analogy again, whether or not the plane's going to go up or down, or stay pregnant or go into labor. So the activity of what happens at this interface, we think is really important. And, clinical data would suggest that chorioamnionitis or inflammation within this choriodecidual interface is one of the high risk factors for preterm birth. Now, the other interesting thing about this interface is that these chorion trophoblast cells are producing bucket loads of progesterone that are acting directly on the adjacent tissues. So this progesterone is affecting the decidual stromal cells and the myometrial cells. So for most of pregnancy, we think that the progesterone is acting on the decidual cells to keep the immune cells under control. And we're looking into different ways that this happens. But the bottom line is that this anti-inflammatory effect of progesterone acting on the PRs in the decidual cells and the myometrial cells dominates for most of pregnancy. But somehow that is overcome. So before we reach that threshold, then this locally produced progesterone acting through the PRs, in this case in the decidual stromal cells, is able to keep the inflammatory status of that maternal fetal interface under control, and everything remains quiescent. But when the threshold is reached, then we think the inflammatory load actually activates or the expression and activity of the aldoketoreductase 1C1 that then prevents the progesterone from interacting with the progesterone receptor and actually inactivates it. So now we have unliganded progesterone receptor, and that has consequences. But what happens is that we lose that ability to maintain a controlled immune state an inflammatory state at the maternal fetal interface. The inflammation then produces prostaglandins that cause contraction of the uterus and dilation of the cervix, and triggers this positive feedback pro-inflammatory state leading to emptying of the uterus. So the work I'm going to tell you about was done by a terrific graduate student in the lab, Angela Di Tommaso, who's just finishing up, and this is some of the data that is being sent off for publication hopefully soon. And so what Angela did was study the fetal membranes that we get from C-section deliveries as explant cultures, so cutting them up in little bits and putting them in a dish with some DMEM and watching them for, in short term, just overnight and possibly up to about 24 hours, or a decidual cells, the decidual stromal cell line that we call IDEC, that can be induced to decidualize by exposing them to forskolin E2 and some progesterone. So in both cases, these model systems are seeing a decent amount of progesterone just under basal conditions, because if you remember, the whole tissue itself has the chorion cells in it. So if we look at the media from these explant cultures, we can actually measure a decent amount of progesterone. Okay, and the IDEC cells are seeing progesterone. So when we look at those tissues, when Angela takes fetal membranes from women at term who are either in labor or not in labor, or at preterm who are in labor or not in labor, we see that the level of expression of the AKR1C1 gene is increased. So that sort of fits our hypothesis that an increased expression of this gene that encodes for something that will inactivate progesterone goes up in association with the onset of labor. It's not telling anything about causality. It's just saying it goes up. And interestingly, our collaborator Suhas Kalpour at UCLA has a beautiful rhesus macaque model where he can take rhesus macaques at 80% gestation and inoculate them with live E. coli into the amniotic fluid. And all these animals go into labor within 24 to 48 hours. And if we look at the fetal membranes from these animals, we also see an increase in AKR1C1 gene expression. So that was good. Well, what happens if Angela takes the IDEC cells or the explants and exposes them to IL-1 beta? Well, again, we see an increase in AKR1C1 in response to the inflammatory challenge. So this is direct evidence that at least with a monolayer of decidual stromal cells on a dish, that they will respond to an inflammatory challenge that we know will drive parturition, IL-1 beta, by expressing AKR1C1, and it also happens in the explants. So our hypothesis, as I said, is that the inflammatory load drives our AKR1C1 that then inactivates progesterone, leaving the progesterone receptor unliganded. But what if we now are able to ligand that progesterone receptor with something that is not susceptible to inactivation? And we can do that with a synthetic progestogen, R5020. And R5020 is synthetic, so it hasn't been seen by nature. And so we haven't evolved an enzymatic activity to inactivate it. So it's the perfect way of bypassing the enzyme. And so what Angela did was expose the tissues in culture and the cells on a dish to R5020 and ask, can we compensate for this enzymatic activity by giving this non-metabolizable progestin? And sure enough, when you take the explants, remember they're already seeing progesterone, and give them IL-1 beta, now we're measuring the expression of a progesterone responsive genes that is in decidual stromal cells, IL-15. There's a whole backstory to IL-15. We think it's involved with regulating the function of NK cells in that microenvironment, and it's responsive to progesterone. So under basal conditions, it will be expressed. If you then expose the tissue to a inflammatory challenge like IL-1 beta, it goes down because progesterone receptor is no longer liganded by the natural ligand. But if we give them R5020, then we can override this inhibition by IL-1 beta. So that was reassuring to us. What really became reassuring was that if we take a mouse model for LPS-induced parturition. So let me explain the model first. So in this diagram, what you see is time on the x-axis, and this is days of pregnancy in the mouse. Mice usually deliver at 19.5. But if we take mice and we give them an intraperitoneal injection of LPS at day 14, most of the animals will go into labor within 24 hours. So here is inflammation-induced parturition. And even if we give them progesterone, okay, they still go into labor within 24 hours. So the green is the progesterone-treated animals, and what we're showing on the x-axis, on the y-axis, is the number of animals that that deliver after 24 hours. And we see here that 23 of the 26 animals that got LPS went into labor. But if we load some of the animals up with R5020 the day before they get LPS, and then challenge them with LPS, and we give them P4 and LPS and R5020, then only 3 of the 26 animals went into labor within 24 hours. And in fact, most of them went on to deliver at term. And so what's R5020 and how can I get my hands on it? Well, R5020 is actually approved for clinical use in France, Portugal, Tunisia, and Argentina. It's sold as Progesterone. It's commonly referred to as Promegestone, but it is used for postmenopausal symptoms and various other clinical uses as a synthetic progestin. And so we published these data, and we're very excited about the potential repurposing of promegestone or something like it as a better progestin to prevent or decrease the risk for infection-associated preterm birth in women. So what I'm getting at is that the progesterone prophylaxis story isn't dead. We know that progesterone works. If you stop it from working, you go into labor. And so what can we do to make it work better? Well, bypassing the natural aldehyde reductase 1C1 with a synthetic progesterone might work. Now, giving natural progesterone won't do it. Okay, so the progesterone vaginal suppositories won't overcome the AKR1C1 gatekeeper. Giving 17-hydroxyprogesterone caproate likely doesn't do it as well for various reasons. It's a good progestin in animal models, but I don't think it's a very good progestogen in women. Okay, so how can we take this to the next level? Well, what Angela did was get a hold of a 20-alpha hydroxysteroid dehydrogenase. That is the enzymatic activity that takes progesterone and inactivates it. That is encoded for by the AKR1C1 gene. There's a compound called BPSA, and I should have written it down because I can't remember what that stands for. Anyhow, salicylic acid, bromophenol silyl, something like that. In any event, this is a potent inhibitor of the 20-alpha HSD activity of AKR1C1. And so we figured, well, maybe we can use this as a tool to dissect out this pathway. And I'll show you one experiment where Angela took the fetal membrane explants and exposed them to IL-1 beta, then measured the response to IL-1 beta by looking at IL-8 expression. And sure enough, she got this nice increase in IL-8 mRNA levels in response to IL-1 beta. But remember, these cells are also this tissue is also producing progesterone. And when she exposed the tissue to BPSA, which presumably inhibits the AKR1C1, so now progesterone is free to act on its receptor, we lost this stimulation by IL-1 beta. Great. But we weren't sure whether or not the BPSA was acting directly through a progesterone-mediated pathway or it's just a natural anti-inflammatory. So what Angela did, clever experiment, was to add RU486 to the mix which is a progesterone receptor antagonist. So now, even though we've freed up the progesterone, binds to the RU486 has a much higher affinity for the progesterone receptor than the natural progesterone, so it'll antagonize this pathway. When this pathway is antagonized, then we see the rebound, or we rescue, in a sense, this IL-1 beta induced IL-8. So that was evidence to us that this may be a way to move forward, in addition to bypassing the AKR1C1 roadblock to progesterone interacting with the receptor. We may be able to inhibit that effect. So,
Great. Sam, just a time check for you. We're within probably the last 5 minutes. So if you want to.
Slide.
Oh, perfect. Okay.
Yeah. So when we give BPSA to a mouse, and remember, this is the same protocol as with the R5020, we see that LPS induces parturition in pretty much all the mice. If we add P4, this should be a plus here, I'm sorry, it still induces parturition here, but when we add BPSA, it rescues this LPS-induced effect, and most of the animals are still pregnant after 24 hours, and the LPS is actually increasing the AKR1C1 in the decidua of these mice. So we're excited about this. We're excited about two potential therapeutics that could be used to boost the actions of the in vivo progesterone. And so I'll end with that, and hopefully I've left a little bit of time to take questions. These are all the people we collaborate with, did all the work, and our funders, the March of Dimes and the NIH. And I really thank you for your attention.
Great, Sam. Thank you so much. Beautiful, beautiful talk. People feel free to come off mute to ask questions or leave a question in the chat.
I have a question, Sam.
Yes.
So what do you feel was the major flaw with the 17p story?
Oh, yeah. Well, I could drill down into that. If you look at the MEIS trial from 2002, one of the things that always made me uncomfortable was that the placebo group had a 50%, you know, and they expected it, that the therapy would, they expected the placebos to be at 30% because it was repeat preterm birth. They had a history of preterm birth. 17-hydroxy-prog treated women were at 34%.
Right.
And if it wasn't for the placebos being at 54%, we would not be giving 17-hydroxyprogesterone caprate.
And it looks like they were also, the placebo group was a higher risk group despite the randomization, and they had to do some corrections afterwards.
Yeah, there was a lot of gymnastics going on there in terms of clinical trials. But then it was repeated in the prolonged trial, and that sort of put the kibosh on the 17-hydroxy-P.
Is there something about the progesterone itself? I mean, you talked about synthetic progesterone versus natural progesterone. Is that the key vehicle?
If you use a synthetic that isn't metabolized, we're proposing that that's a better way to overrun. I mean, there's probably there's redundancy in progesterone withdrawal. Okay. All pregnancies must end. And evolution would have built a redundancy in order to save mom's life in case the ending of trigger mechanism for the ending of pregnancy goes awry. And so one of the mechanisms we think is the metabolism of progesterone. And so if you can override that, at least that form of progesterone withdrawal has been fixed. Whether or not you want to do it is another question, because do you necessarily want to, if you don't know the wellness of the fetus, do you necessarily want to block all the progesterone withdrawal mechanisms? And
Sam,
so,
that actually ties beautifully into my question in terms of, you know, infection, intrauterine infection, we may not be wanting to block. So the model in the mouse that you're utilizing is an infection-based one.
exactly.
In terms of sterile inflammation, because we think, inflammation without infection probably is driving the parturition at term and could
Right.
account for some preterm. Do you know if the AKR1C1, the pathways that you're looking at, are there differences whether you have a sterile inflammation setup or an infectious
Yeah,
stimulus?
that's something we're looking at with Suhas Kalpour with his rhesus model. And so the rhesus model that he's got going is you cause an infection, intrauterine infection, and then within 24 hours, you treat with antibiotics. So what he does is he clears the infection. There's no septicemia in the mom. If you don't do that, the moms all get septic and they all have really hard time. And the amniotic fluid is just a cesspool. But if you treat with antibiotics within 24 hours of inoculating the amniotic fluid, then these animals have no signs of intrauterine or maternal infection, yet they all go into labor. All right. So what we think is happening is that, once you set off this positive feedback loop of tissue level inflammation and progesterone withdrawal that begets more tissue level inflammation, feedback loops are really tough to interrupt unless you get ahead of them, because the only way to interrupt a positive feedback loop is to, in this case, shed the tissue where the loop is happening, and that's the choriodecidual interface. What I'm getting at is that it's possible, if you can detect an intrauterine infection, to treat with antibiotics and then treat with a R5020 or an AKR1C1 inhibitor downstream of that to try and stop the 747 or the 787 now barreling down the runway.
Right.
I don't know.
All right. Well, we are over time. And so I would like to thank everybody for coming, and for Sam for giving such a clear and lovely talk. I think even for the non-basic science in the audience, you did a beautiful job of telling the story so that it could be understood, which is much appreciated. And I wish everybody a happy holiday, safe travels if you're traveling. I hope everybody stays well.
Virginia, one quick thing. Deirdre
Yes.
had her hand up. If it's a quick question, Deirdre.
It's not. I mean, we're past time, but
Okay.
just more about etiology of preterm birth and PRA versus PRB levels. So
Maybe offline with Sam.
Well,
Yeah.
I'm happy to stay on if you want to.
Okay, why don't we
that'd be
do that?
perfect.
So if
Yeah.
people have to
Okay.
run, feel free to jump off. And for those that can stay and have additional questions, feel free to stay on.
Thanks so much, Dr. Mesiano. It was fantastic.
Oh, thank you.
Yeah, thank you. That was wonderful. Yeah, so my question is more just about etiology, and that was really wonderful and easy to follow for a a clinician, non-scientist like me, non-basic scientist. I'm curious as to, not all preterm labor is inflammatory or infectious in origin, and so thinking about why some people just develop it versus others. Like, has anybody looked at, say, differences in PR? Like, do some people have more PRA like receptors than PRB? Are just levels then, like, has anyone examined those? Like, does an
The short
etiology?
answer is no. And the slightly not shorter answer is that now that we know more about the molecular biology of the progesterone receptors, I don't talk about the PRA, PRB story much anymore, because every time we do something in the lab, we get data that makes us rethink. I'm not saying the original data is wrong, but now we're getting into the real weeds of how these receptors are interacting with each other. And we kind of think now that the Western blots that we did originally could be done with a bit more resolution, if you will, and that the AB story isn't as straightforward as we think, because there's now a phosphorylation component to it.
Mhm.
We know that it's infection IL-1 beta on a dish and LPS on a dish that will drive the A effect, but it's really tough to get the tissue for one, and have it be from and understand the phenotype of the woman that you're getting the tissue from. It's not just in labor, not in labor. It's, as you guys know, there's so many different reasons why a woman might be having a C-section other than scheduled. And so I recoil a little in making dogmatic comments about PRA and PRB. And it was my data, so sort of I have the privilege of doing that.
Well, that's helpful. Thank you.
I should be the one doing it.
Yeah, actually, I had a question about the two isoforms. So it's the same gene. So it's exactly the same sequence except for the first domain, the BUS domain. Is that correct?
Yes.
And the phosphorylation site. So then I thought there was an implication with how you presented it that there is differential phosphorylation of A and B. And I'm curious as to how that is, given that the sequence is the same and the phosphorylation is the same.
Virginia easily gets in the weeds, and that's a whole bunch of weeds right there that you actually don't know where you are when you're in it. It's like roaming around in a forest. But in vivo, if we take pieces of tissue from women having a C-section, the ones that are in labor will all show phosphorylation only on PRA and not on PRB. And
That's so crazy. Okay.
We could not explain that. And now what we think might be happening is that B is being proteolytically cleaved once it becomes phosphorylated to something that B is a pro-protein. It can be cut up, and we have evidence.
And something about that other domain is guiding whether it gets degraded or not degraded when it's phosphorylated.
The B upstream segment has got lots of it's got one critical activation factor on it that is necessary for PRB to regulate the transcription of a whole set of genes. And most of the people studying PR study PRB because it's transcriptionally. If you cut that thing off, it turns into PRA. And it does completely different things. And what we're seeing on our cells on a dish, we've got evidence that it's being cut off, that the damn thing. And so this is why we don't know whether this is happening in tissue we get from women, but what if it is? Then it puts a whole new twist, because it'll make the band that we called PRA
PRB look like PRA.
Yeah, it's not PRA increasing, it's PRA, but PRB being metabolized. And so when we look closer at the Westerns, we start thinking, well, that's the band we could never explain. And now we're sort of circling back and backpedaling. And this is science. For those of you who don't do basic science, it's a little puzzle that happens, and you've got to try and fit your Tetris pieces into these holes that are changing their shape all the time.
Yeah. Wonderful. Well, great.
Hi, Sam. It's Cece. I have a quick question. I'm just wondering if there's any evidence, Sam, to suggest that there's like a differential effect of inflammation at different gestational ages or at different progesterone receptors, because, choreo in, preterm birth, choreo triggers labor, but then, we have all these term laboring patients who get choreo and then they get dysfunctional labor. And then even, sort of on the same lines, like the R5020 data that you showed in mice, right? Those mice still went into labor. It's not like you prolonged gestation either.
Well. That's a great question, because the original study that was done by our collaborators in Toronto, Steve Ly's group. They gave a massive amount of R5020 to these mice, so much so that they didn't even go into term labor. It hung
really?
around for, and they basically had to kill the
Mm.
mice in 23 days. When we pulled back on the dose, we could see that now animals would get over this. And again, it's what's happening in the rhesus as well. You have this inflammatory challenge, which Suhas gives live E. coli into the amniotic fluid. That will trigger an event that says, oh, I must empty the uterus. And even if you get over that inflammatory challenge, like LPS is not infectious, it's just yes. So that's cleared out. But because you've triggered the positive feedback loop, then the uterus gets emptied. Now, what's happening in women? That's your job to figure out, in a sense, because every woman's different, and it could be that distension is, myometrial distension is playing a role. It could be that the vaginal microbiome is, and the cervicovaginal fluid changes are sending signals up through the cervical os that might be contributing. What I'm getting at is that there are possibly multiple factors that contribute to this inflammatory load trajectory. I showed it as a straight sort of smooth curve, but it might have spikes in it that happen. We don't know what the shape of that curve is. We simplify it so we get it, that we can get our head around a model that we can at least poke around at in the lab. But to me, it makes sense that there's redundancy in the pathways to end pregnancy. What is always been interesting to me as well is that there's not a lot of redundancy in the pathways that maintain pregnancy. So all we have is progesterone. If you kill progesterone with either taking the progesterone away, overreactomizing a mouse, or giving RU486 to any species, pregnancy ends. So natural selection hasn't built in any redundancy into getting pregnant, but it has built redundancy into ending pregnancy. The problem we have as humans is that the redundancy that was built to end pregnancy, I think, and I have a whole set of slides on this, is in what affected that was, our natural history. We became bipedal and we encephalized. So we've got to get this big head at the obstetric dilemma. So how do you deliver these very immature altricial neonates? Whereas if you compare us to the rhesus or the baboon, other primates on the planet are delivering toddlers. We're delivering pinkies. We're essentially mice. And so something happened in our evolution to change the trigger mechanisms for parturition, and probably maybe we're hyper responsive to any inflammatory or stressor. The benefits of that is we empty the uterus before the head gets bigger than the hole. The cost of it is that we have an increased risk for preterm birth. But who cares? Natural selection doesn't care about the costs as long as the benefits outweigh the costs. And so I could rant on about that forever.
Yeah. All right.
Thank you.
Okay. Well, if there's no further questions, again, wish everybody who's still on a happy holiday. And thank you, Sam.
Thanks, Virginia. Nice seeing
I'll
you.
see you at SRI and we'll talk more.
Vancouver.
Yeah.
Right. Thanks a lot, everyone.